All right, good morning everyone. We're finishing off with our osmosis design part today. So hopefully by the time that you're watching this, you will have deconstructed, which is part B, the problem around how to measure and test the movement of water in and out of single-celled organisms, or unicellular organisms. Whereas now you're moving on to part C, which is where you design a practical investigation looking at the movement of water, which is osmosis, in potato cells. So our learning intention today is to design a biological investigation to measure the impact of one factor on osmosis. So I want you to think about what factors affect osmosis. We have looked at least 3 of them in class last week, one of which I actually demonstrated. Some of you are quite amazed or just thought it was quite pretty the way that it mixed together. So if we have a think about that, there are the salt concentration. So you would have looked at the amount of salt relative in both the interior and the exterior environment, and that can create different types of solutions: hypertonic, hypotonic, and isotonic. If you are a little bit unsure of those terms, please go back to the PowerPoint from last week, which looks at whether if you have a hypotonic environment, which way the water is moving into the cell or out of it, and vice versa for a hypertonic solution. We know, given the demonstration that I gave last week, that the amount of osmosis that can occur is affected by temperature. And we've also got surface area to volume ratio as well, which is basically how much surface is available in which water molecules can move in or out of a cell. So for your part C, you need to design an investigation which, as I said, tests one effect of one factor, which is your variable, which in this case your independent variable, on the amount of osmosis in potato cells. So the way that you're going to measure osmosis is by weighing your potato cubes. So you're going to cut your potato into cubes. Before and after a set amount of time, I would recommend at the very least 20 minutes, if not longer, to calculate the difference in weight, which will tell you how much osmosis has occurred. You will complete multiple trials to ensure that you have precise results, and this needs to be completed, the design, by the end of this lesson. You will then conduct the experiment Monday next week. Again, it's very important that if your design is not clear, and as a result the lab tech does not provide you with all the equipment that you've asked for, this will affect your ability to complete your practical and also your grade as well, because I am grading you on your ability to design a biological investigation. So you're probably wondering, what do I need to include? You need to include a hypothesis. This is an if-then statement. So for example, if you were looking at temperature, if I increase the temperature of the water, then more osmosis will occur. Okay, that's testable. We're varying the temperature and we're recording the weight difference in the potato cubes, which should indicate movement, either loss of water or gain of water. You need to include the aim. What's your aim? What are you trying to actually do? What's the outcome that you're looking for? You need to include your independent, which is the variable that you are deliberately modifying, temperature, surface area to volume ratio, or salt concentration. The dependent variable, which is how you're going to measure it, I've already talked about that, and your controlled variables. So obviously everything else in this experiment has to stay the same so that we can say without a doubt that the surface area, for example, was the only thing that could have impacted the amount of osmosis. Because if we don't have the same temperature, and if we don't have the same potato breed, or if we don't have the same amount of time in which we keep our potatoes in there, or the same way that we're going to measure them, that's going to introduce errors and make the conclusions that we want to make a little bit more unreliable, which we don't want. You also need to include a materials list. Okay, I'm going to go through in a minute what an A looks like for a design, so you have an idea of the kind of detail that you need to go into. You need to include a method. It's your set of instructions. And you need to include a risk assessment looking at both safety and ethical considerations. Okay, so what does an A look like? If we have a look here, this is from another investigation looking at the ratio of dilution that's needed of local bore water. So bore water is groundwater. That tends to be a bit salty in order to successfully grow lettuce. Okay, so this student, this was actually for a different investigation. They had really salty bore water that they wanted to use at their house to grow lettuce. But when they realized that if they just used the bore water by itself, the lettuce wouldn't grow. So they're trying to work out how much water they need to dilute the bore water by, so fresh water, in order to allow lettuce to grow. So that's a specific question. You could write this as a hypothesis as if the dilution of bore water were to increase, then the amount of growth seen in lettuces would increase. Okay? If-then statement. So in terms of the independent variable, it's the ratio of rainwater to bore water. Okay, that's what we're changing. The dependent is the size of the lettuce plant. Okay, and the student actually measured the length. The controlled variables include the breed of lettuce, so it has to be the same breed. The pot size in which the lettuce is growing, the amount of water that it's actually given, the amount of sun, the soil type, the amount of soil, time allowed to grow. These are all factors that will impact the growth of a lettuce plant, which means that they all need to stay the same so that we know that the amount of salt in the water is the only thing that is impacting the growth of the lettuce. Okay, so if we move on to our materials list, you'll see it's quite extensive. We've got the dimensions of the items included. We've got the number of items that are included. We even have resolution, which is like centimeters versus millimeters, on certain items. It needs to be a very specific list. Moving on to the procedure, we can see that there are specific detailed instructions with even further instructions for mixing the bore and rainwater ratios with a table. So it's really clear how to actually measure out your different ratios. So if you're looking at salt concentration of water, perhaps you want to adapt something like this for your investigation to make it very clear. The whole point of a good design is that I should be able to pick it up, follow your design and come up with the exact same results as you because it's that clear. So if your instructions are a bit vague or very basic, chances are I will not complete the investigation or the design in the same way that you did because the instructions aren't detailed enough. Okay, you also need to include a risk assessment. I do recommend that you include this as a table. It's a really nice way to actually lay out and give all the types of risks. So there is the risk, why it's a risk, what prevention needs to happen, and if the risk were to occur, what treatment or action needs to occur. So a detailed explanation of the risk, a justification of why it's a risk, the type of risk also, and specific instructions about how to prevent, and a detailed plan if the risk were to occur. All right, so your submission of your design, as I said earlier, is today, Thursday, via Daymap. The assignment's already up there. It needs to obviously be written up as a Word document. I don't have a template for this. You can just write it up using. These are what you need to include, okay? And use this obvious example here as a way to make sure you've included everything. Excuse me. I will be assessing you on your ability to design an investigation, okay? So I will be looking to see, have you included all of these different aspects? Do you have a specific hypothesis? Have you identified all your different variables? Do you have all the materials needed, and are they specific? Do they have dimensions? The number? Your procedure, can I follow it? Is it ambiguous? Have you got further instructions? I should be able to pick this up, not needing any prior knowledge about your investigation, and should be able to do it just like you did. Okay, if you have any questions about that, you can email me. I will be on my computer, like I said yesterday, during your lesson time. I'm just not physically there, so you can ask the questions if you need to. Good luck.